welcome everyone. We're very excited to be hosting this webinar today, looking at the use of NLP to improve phenotype extraction for precision medicine. I'm joined today by Benjamin Darbo, MD, PhD, who is the Assistant Professor of Pediatrics at the Stead Family Department of Pediatrics at the University of Iowa, and also by Alyssa Hahn, who is a doctoral student in the interdisciplinary graduate program in genetics, also at the University of Iowa. We will be looking at a few introductory slides just to explain some of the NLP concepts and give an introduction to those ideas. And then I'll be handing over to Ben and Alyssa to explain the work that they've been doing using the Linguamatics NLP platform. Really fascinating work in computational phenotyping and a really exciting area in the precision medicine space. A few introductory remarks about Linguamatics. We are an IQVIA company, which is the merger of IMS Health and Quintiles. Been working in the NLP space for around 18 years, focused in the application of NLP to healthcare and life science applications, addressing a wide variety of different sources, and really focusing in on the concepts of extracting complex disease concepts, medication concepts, adverse events concepts from electronic health records, from scientific literature, from clinical trials, definitions, and that's the focus of what we do. And as we consider the space within which we work in healthcare and life sciences, there is so much unstructured data for us to be able to analyze and to be able to extract insights and information from. It's very hard to be able to keep up to date with all the scientific literature, to be able to read through the clinical documentation and quickly find the answers that you're looking for. And that's really where NLP comes in terms of being able to computationally read through different documentation, find the items of interest, summarize that information. And that could be in the discovery and development side of a life sciences company, looking at targets for bone cancer, companies who are patenting particular technologies, looking at drug safety risks, finding the best sites for clinical trials, looking at regulatory approvals, for example, looking at sources like Medline, FDA drug labels, clinicaltrials.gov, for example, as sources of information. And then on the healthcare side, mining electronic health records, looking at patient safety issues, for example, looking at quality measure submissions for regulatory support, decision support, supporting population health, where we're interested in potential social determinants of health that are trapped in clinical notes. And obviously the area we're looking at today, which is computational phenotyping and extracting phenotypic information from EHR data to support the improved diagnostic yield of genetic testing. So when we're thinking about how the NLP process is working, we're taking unstructured documents, a Medline abstract, an EHR patient record. We're processing that through the NLP engine using a variety of different technologies and capabilities, but we're essentially using ontologies like the human phenotype ontology that we'll be looking at in the session today, using statistical methods, machine learning approaches, regular expressions to identify key concepts in the text and then to extract them from the text and normalize them, but also to understand the context in which these concepts have been identified. We can see here examples of ejection fraction. There's a value being extracted from electronic medical records. Each row here represents an individual patient, and we're extracting a value from the text. We're looking at the ranges, but we're also identifying that this measurement was either estimated or calculated, for example. So we're understanding that context for those particular concepts. And that becomes really important as we're considering the different concepts we might be interested in. We can see here different words with the same meaning, often ontology driven. So this would be drugs, diseases, where we're finding concepts that are called different things, but we want to normalize them together to improve the consistency of our analysis. We may see different grammar for the same meaning. We can see here different cyclosporin dosages in terms of volume and frequency, and how they're written is variable, but the information is the same. So again, we need to be able to normalize that. For non-smoking classification, for example, there might be lots of different ways that would be written within clinical notes. Again, we're looking at how we normalize that information and make it consistent. And then really importantly, and this will be a strong theme within the rest of the presentation, is looking at the context. Is this diabetes term something that was diagnosed? Is it a family history? Is there no family history? So negation and context are really key when we're analyzing and looking for these data and these concepts within documents, because it's not just finding the word, it's understanding how the word is being used.